The human body is operated by millions of microscopic machines. Proteins are massive molecules that handle everything from digesting meals to neutralizing viruses. To design effective medicines, we have to understand how these machines function, and that requires knowing their exact weight and three-dimensional shape. For most of the 20th century, mass spectrometry was a violent process. Early machines used intense heat to turn molecules into gas for measurement. While heat vaporizes small molecules easily, proteins are different. Chemists call them molecular elephants because of their sheer size. Applying enough heat to vaporize these giants instantly tore their delicate structures into useless, disconnected particles. Scientists were stuck in a cycle where the very act of looking closely at these proteins destroyed the evidence they needed to see. The breakthrough that earned the 2002 Nobel Prize in Chemistry was the realization that these molecules didn't need more power. They needed a gentle touch to safely lift them into a vacuum. American chemist John Fenn hypothesized that he could isolate proteins by using an electrified mist instead of heat. In Fenn's electrospray method, proteins are dissolved in liquid and sprayed through a charged needle. As a counterflow of gas evaporates the water, the droplet shrinks until only the intact charged protein remains hovering in the air. At the same time, an engineer in Japan named Koichi Tanaka tried using lasers to solve the vaporization problem. During his experiments, Tanaka accidentally mixed glycerin into a sample suspension. Rather than discarding the ruined mixture, he decided to fire his laser at it to see what would happen. The glycerin acted as a matrix. It absorbed the laser's destructive energy and vaporized, acting as a cushion that launched the target protein into the detector without breaking it. This graph provided the proof. That peak represents a completely intact macromolecule hitting the detector, demonstrating that it had survived the ionization process. By solving the vaporization problem, Fenn and Tanaka gave scientists a way to accurately weigh and catalog every intact protein in the human body. A precise weight identifies a protein, but it doesn't reveal how it functions. To see how these machines actually work, researchers have to map their three-dimensional architecture. The standard mapping method, X-ray crystallography, required freezing proteins into solid crystals. It produced high-resolution maps, but it stripped the proteins of their natural movement. Kurt Wüthrich wanted to observe proteins as they exist in living systems, swimming freely in water. He turned to nuclear magnetic resonance, or NMR. While others used it for small molecules, Wüthrich envisioned a way to adapt the technology for massive biological structures. By placing samples inside powerful magnets, he used radio frequency pulses to calculate the exact distances between individual atoms within a floating, flexible protein. Computers then use these thousands of distance measurements to render a complete map of the protein's folded structure. Today, high-resolution NMR analysis allows scientists to study molecular interactions under conditions that closely mimic our own body fluids. This liquid mapping proved that proteins aren't static objects. They are dynamic machines that shift their shape to perform their duties in our blood and cells. These breakthroughs in weighing and mapping molecules have allowed us to track and respond to lethal diseases with high precision. For example, Wüthrich's NMR allowed researchers to map the specific misfolded proteins, called prions, responsible for neurodegenerative conditions like mad cow disease. In drug development, scientists can now observe how small medicinal molecules physically bind to a giant protein receptor without damaging the target. These three laureates established the field of proteomics. They provided the tools to catalog and study the physical products of the entire human genome. By developing this gentle approach to chemical analysis, we finally gained the ability to see and weigh the physical molecules that make life possible.